Hello everyone, it's a pleasure to share with you the latest results from our team. MicroRNA-microbiota interactions, emerging strategies for modulating intestinal homeostasis and enhancing host health. Long-term artificial selection and environmental shifts have driven adaptive changes in both the host genome and the intestinal microbiota. The complex symbiotic relationship between these two has become essential for maintaining intestinal homeostasis and overall health. Concurrently, advancements in sequencing technology and the functional annotation of non-coding RNAs, particularly microRNA, miRNAs, have facilitated the exploration of mechanisms regulating intestinal homeostasis. Herein, we systematically update the role of miRNA-microbiota interactions in regulating the intestinal barrier, intestinal immunity, changes in intestinal microbiota dynamics, and maintenance of intestinal homeostasis, and we further critically discuss the role of miRNA-microbiota interactions in the maintenance of host intestinal health, metabolic regularity, brain function, and neurodegenerative disease-related disorders. Finally, we highlight the prospects and therapeutic strategies regarding miRNA-microbiota interactions in humans and animals in the context of intestinal microbiota and gene function studies. Within the intestinal, miRNAs are primarily produced by intestinal epithelial cell, IEC, and HOP homeobox, HOPX, positive cells. Importantly, the types and abundance of miRNAs within this environment are not fixed, and rather, they exhibit considerable variability. In addition, there are complex interactions between miRNAs and microorganisms in the gut, and these reciprocal effects would be further involved in gut homeostasis construction. miRNAs are extremely abundantly expressed in the intestinal mucus layer and IEC and modulate the reception of signals from commensal microbiota or invasive pathogens by IEC that subsequently adapt to altered intestinal environments by modulating the mucosal barrier and signaling to immune cells in the lamina propria. This suggests that miRNAs are not only important for regulating the integrity of the intestinal barrier, but are also key inflammatory factors in the intestine and markers of the intestinal inflammatory cascade response. In inflammatory bowel disease, IBD, including Crohn's disease, CD, and ulcerative colitis, the dynamics of a number of these mRNAs can directly affect immune cell maturation, differentiation, and infiltration, the intestinal immune system, and intestinal homeostasis in conjunction with target chemokines. Autophagy of IEC is an important mechanism for removing harmful intestinal substances, regulating the intestinal immune response, and maintaining the intestinal barrier and intestinal homeostasis. miRNA microbiota interactions can be involved in the autophagy process of IEC in various manners, including target binding of mRNAs to autophagy genes, regulation of endoplasmic reticulum protein synthesis, and modulation of intestinal immune pathways. The bidirectional neurohumoral communication system between the intestines and the brain is known as the intestinal microbiota, brain axis, and studies have confirmed that this bidirectional communication may be closely related to the body's metabolism, endocrine and immune activities, neurological development, and intestinal homeostasis. In recent years, increasing evidence has demonstrated that intestinal flora and miRNAs can regulate brain development and function through the intestinal brain axis, and this in turn affects host brain function and behavior, as mRNA microbiota interactions are not only manifested in the regulation of intestinal health, organismal inflammation, and nutrient metabolism, but also in the modulation of the synthesis of neurotransmitters and the development of the nervous system. Therefore, miRNA microbiota interactions play important cross-regulatory roles in body metabolism, intestinal immunity, intestinal homeostasis, and microbiota, intestinal, brain axis-related diseases. With the support of sequencing technology to sequence and analyze mRNAs and microbiota from different groups and different health states, the correlations among them are beginning to be revealed, and their regulatory mechanisms in the context of intestinal homeostasis can be further investigated. Meanwhile, based on the approach of multi-omics joint analysis, the integrated study of miRNA microbiota interactions with host metabolism and immunity is anticipated to reveal individual differences and the mechanisms underlying related diseases. Meanwhile, the regulation tools for the microbiota will be more diversified, including probiotics and prebiotics, and others, to allow for precise intervention against the microbiota. This study summarizes the regulatory mechanisms and modes of action by which miRNAs interact with the microbiota to regulate intestinal homeostasis and host health. It is evident that the use of miRNA microbiota interactions for personalized regulation of intestinal homeostasis possesses great prospects and potential. This research direction will not only deepen the understanding of intestinal microbiota ecosystems and miRNA regulatory networks, but also promise new hope for personalized medicine and the treatment of intestinal-related diseases. Thank you.